Dr. Vaidevi, shall we start?
Yes, sir. I will start, sir.
So, good morning, everyone. So, on behalf of Society of Chemical and Synthetic Biology, myself Surya, welcoming all. Today I am introducing Dr. S. Vaidevi. So, she is a young scientist, so awarded Department of Health Research, Government of India, and also she is the research head of Ratnam Technical Campus, Coimbatore. So she was selected for Young Research Scheme with a worth of 51 lakhs project for a period of 3 years of 2022 to 2025 under the Department of Health Research, Ministry of Health and Family Welfare, Government of India. She was awarded for the doctoral degree of in 2021 under Anna University, Chennai for a research work in the area of drug delivery applications for lung cancer. And also she was awarded for ICMR Senior Research Fellow for the period of 3 years during her doctoral studies. She has the experience of 5 years in research and she has published more than 11 papers in well-reputed international and national journals with a cumulative impact factor of 36. So with this, I will welcoming Dr. S. Vaidevi. So it's over to you, Vaidevi. Please.
Yes, sir. Thank you, sir. Thank you for your invitation on my guest lecture topic, Nanobio Foundation for Drug Delivery Applications by the Society of Chemical and Synthetic Biology. Hereby, I welcome all students, staffs, and research scholars, my colleagues, for my guest lecture presentation. Myself, Dr. S. Vaidevi. Then, before going to the topic, I would like to tell about my area of specialization. My main area of specialization is nano drug delivery systems, specifically for cancer therapeutics. And I have well-established knowledge, training and knowledge in chromatographic techniques such as HPLC, LCMS, and HPTLC. So these are my area of specialization. My area mainly focused on the drug delivery applications. And I have a research experience as a senior research fellow in ICMR, and also I worked as a project assistant. And currently I'm working as a young scientist in Ratnam Technical Campus and head research for the Ratnam Technical Campus, and I have a cumulative impact factor about 36.14. These are my Google Scholar and LinkedIn profiles. And next, coming towards my funded research projects. The current ongoing project is development evaluation of bevacizumab nano dispersion for effective treatment of lung cancer. This is a drug. Its trade name is Avastin. And this project was funded by a Department of Health Research, about 51 lakhs for the period of 3 years, under the mentorship of Dr. B. Nagaraj, Principal and Professor, Department of Biomedical Engineering, Ratnam Technical Campus. This is a current ongoing project I am carrying out. Then I have finished one of the ICMR Senior Research Fellow projects. It's also based on the drug delivery applications only. Resveratrol Costin, drug-loaded nanoparticles itself for the colon cancer. This is a research work I have completed in 3 years, June 2018 to 21, under the guidance of Dr. K. Rukmani, Professor, Department of Architecture Technology, Anna University, Tristapalli. These are the funded research projects. Apart from these funded research projects, I have carried out some other projects for my studies. First one is doctoral research work. Actually, my PhD work is also based on the drug delivery applications only. So the session will be more informative based on my PhD work. My title of my PhD work is pH responsive nanoparticulate drug delivery system for the effective treatment of lung cancer. So, and this work I was done under the guidance of Dr. K. Rukmani, Professor, Anna University, Tristapalli. After that, before my PhD works, I joined as a project assistant under the some project called DST Supported National Facility for Drug Development. In that, I have carried out one research work. That work also based on drug delivery applications only. That work is pH responsive metformin loaded zeolitic imidazole framework for the treatment of lung cancer. And these are my UG and PG research works, that is a BTech and MTech research works. PG research work was I carried out in Nanobio Foundation for Environmental Applications under PH Institute of Advanced Studies. And the UG research work I carried out in enzyme production under the Department of Biotechnology, Anna University, Tristapalli. Then coming to our today guest lecture topic, nano drug delivery systems. First, we should know about the nano drug delivery systems. What is the need for that? That's the main theme of the today presentation. Lot of drug delivery systems are available, but all drug delivery systems contain some disadvantages and demerits. To overcome this conventional therapy, we need effective treatment methods. For that, we are going for nano. My area of specialization is mostly in cancer, so I focus on the cancer only. So cancer is already we all know this proliferative disease, that is a uncontrollable growth of cells and tissues in a particular region or a particular organ, that's called as a cancer. In cancer, there are a lot of conventional treatment options are available till now, that is surgery, chemotherapy and radiotherapy, bone marrow transplant and immunotherapy. These are the conventional treatment options available, but these treatment options are having a lot of side effects. That is mainly it is having a poor therapeutic efficacy. We need to give a more amount of drug for the patients, and non-specific interactions. It is not site specific to the particular cancer. It will be kill the normal cells also, so it will lead to the some of the weight loss or hair fall, like that things. So we need a effective delivery system to overcome these challenges. For that, we need a nano. So and also it is a toxicity to normal tissues or cells, so it can't be used for the long time for treatment. So we need a active delivery system which could be a target specific, and it could be a less we have to use for less concentration, like that lot of parameters are there. So we need a nano type of delivery systems. So what is the main need for nano delivery system? It is able to deliver drugs to the target tissues, and the main advantage is we can deliver a drug in a controlled manner. That is the main thing. For example, for diabetes, we have to give a drug for a prolonged time. For that, we can design a controlled delivery system, and the product we are manufacturing or developing can be biodegradable, and it can be easily eliminated from the body after the process gets over. So these are the main advantages why we are going for nano drug delivery systems. Then it contains the two main categories. First one is inorganic nanoparticles, another one is organic nanoparticles. In category of inorganic nanoparticles, most of the metal-based nanoparticles will become, that is like silica, gold, quantum dots, and nanoseria, iron oxide, zinc oxide. Lot of metal-based nanoparticles will come under the category of inorganic nanoparticles. In kind of organic nanoparticles, it is mostly polymeric or lipid-based. So this category is under dendrimers, polymeric nanoparticles, lipid nanoparticles, and polymeric liposomes. These are the organic nanoparticles. Hereby, my research area is focused mainly on both, that is inorganic and organic. In case of organic, I used a specialized nano carrier. I will explain that later. In case of inorganic also, I have used one type of nano carrier. That also I will explain in forthcoming slides. So these are the different type of nanoparticles which can be used for the research purpose. It can be a nanoparticle. We can conjugate our drug by using a specialized conjugation agents. So the main these are the overall main nanoparticles available for the drug delivery applications. First one is a solid lipid nanoparticle. Second one is a nanoliposomes and dendrimers and silica gold nanocell and gold nanocell. These are some of the currently available nanoparticles for the drug delivery approaches. Hereby, my area is focused on mainly is the cubosomes. Cubosomes that comes under the lipid category of drug carriers. It contains a bicontinuous phase. Bicontinuous means it is both water and lipid phase is available. So hereby we can use both type of drugs, that is hydrophobic drugs and hydrophilic drugs. Both drugs we can be able to accommodate within the cubosomes. So it is a lot of advantages. We can use lot of different types of drugs for different types of applications. So my area of theme is cubosomes. Another type of carrier which I have used is a metal organic frameworks. That is another separate work. Metal organic frameworks is comes under the category of inorganic and organic nanoparticle. It contains both the organic ligand and the metal material. So it forms a porous structure. In this porous structure, we can accommodate large amount of drug due to the high surface to volume ratio. So these two carriers which I have chosen for my overall research works. First, we will be move with the cubosomes. So in cubosomes category, I have used the lung cancer. The disease which I have focused is a lung cancer. As already we all know, the lung cancer statistics, it is a predominant disease which affects more women and men, and it will leads to the high death rate in America, and also WHO report, that is World Health Organization reports also told that after breast cancer, the lung cancer is a predominant cancer which causes a high amount of mortality with a high amount of death. So I have focused my research towards lung cancer. In lung cancer, there are two types. One is about the non-small cell lung cancer, another one is a small cell lung cancer. But the non-small cell lung cancer accounts the main 80 to 80% of lung cancer. So focusing my research to non-small cell lung cancer will be better for society and will be better for a better therapeutic platform. So I have focused to the non-small cell lung cancer. The conventional treatment options available for lung cancer is surgery, radiotherapy, and stem cell transplantations, and gene therapy, chemotherapeutics. These are the options available, but we need but these are not site specific. It will have some side effects. So to overcome the side effects, I developed the nanoparticle, which can be a target specific. So for the target specific approach, lot of parameters is there. First one is a responsive method. What is mean by responsive method? The responsive method means at particular response conditions only, the drug will be released and it will be reached the target site. So the major types of responsive systems are pH responsive and temperature responsive, light responsive, enzyme, redox, and magnetic responsive. It means what is the role of pH responsive in cancer? Because many of the students not aware about much about cancer. So I will explain the role of pH in pH responsive, because normal cells contains physiological pH, normal cells, but in case of cancer cells, the pH level in the cancer cell will be in a slightly acidic range, that is less than 6 or 5. So this pH responsive area is which I have focused for my PhD research work. So a lot of responsive systems are available. Here the pH responsive material which I have chosen is a calcium phosphate that I will be explained later. So what is the role of calcium phosphate in lung cancer that we have to know? Calcium phosphate is comes under the type of metal category. It has a high biocompatibility and biodegradability and higher drug loading efficiency. But here, which I have used in calcium nanoparticle, which is mainly for the purpose of pH sensitive property, because of the one of the specialized action called calcium phosphate remains as such in case of physiological pH, that is at pH that calcium phosphate will be remain as such. But in case of acidic pH, that is acidic environmental pH 4 to 6.5, it will be dissociated into ions, that is calcium 2 plus and phosphate. So it will be beneficial for my research work. That is once the calcium phosphate dissociated, the drug release rate will be higher. That I will be explained by using the flow chart and develop the formulation. So the main carrier which I have used is cubosome, and the main pH responsive material which I have used is calcium phosphate nanoparticles. So these are the advantages of cubosomes and calcium phosphate nanoparticles. Cubosome, as I already explained, it is a bicontinuous lipid phase nanoparticles which contains a higher amount of you can due to the higher surface area, we can load a lot of drugs. And second category main thing is it can accommodate both lipophilic, hydrophilic, and amphiphilic drugs. So this is the advantage of cubosomes. Next comes to under the calcium phosphate nanoparticles. As I already explained, here I have employed for its pH sensitive property. So this is my overall PhD work, research work. That is the drug which I have chosen for my lung cancer is a lumefantrine. Lumefantrine is a type of drug. It comes under the anti-malarial drug. So I have used this anti-malarial drug for the treatment of cancer. That is the method is called drug repurposing. That is already available drug for particular treatment. We can repurpose for another disease. That's called drug repurposing. So I have used lumefantrine drug for the repurposing purpose of lung cancer. So the drug which I have chosen is lumefantrine. That calcium phosphate is nanoparticle is used for a pH sensitive property, and the product which I have formed is cubosomes. That is a lumefantrine loaded calcium phosphate loaded cubosomes. So the method which I have employed is lipid emulsification, and the excipients and the other agents which I have used is polaxamer and cetyl palmitate. These are the excipients which forms a cubosome structure under specialized conditions. So this is the overall flow chart of cubosome preparation. After the formation of cubosome, it will be in a liquid form. We have to check for its testing efficiency in terms of characteristics, particle size, and the loading efficiency and pH sensitivity. These are the things we have to check to prove its efficacy. So first I have checked with the particle size. The particle size should be in the nano, or because we are focusing in the nanoparticle database system, the particle size should be in the range of nano. So first I have checked the particle size by using a particle size analyzer. This is an instrument which measures the amount and the diameter and how much amount of particles present in the sample. So this is a particle size analysis. It shows 137 nanometer in size of cubosomes, and zeta potential is a parameter which can be used for the measuring the charge of the particle. That is what is the charge, whether it is positive or negative, that we can be measured by using the zeta potential analysis. Here the zeta potential was found to be minus 28 MV. After the preliminary characterization of size, I have go for a HR-TEM. HR-TEM is a high resolution transmission electron microscope. So in HR-TEM also I confirm formation of nanoparticles within the nano range. So that way it confirms the formation of particle. After that, we have to move for the pH responsive property. We have confirmed the size. So we have to check for its pH sensitive property. So for that, we have to prepare a sample, and we have to analyze for a pH sensitive or different pH ranges. So we can prepare samples with the pH 7, 4, and 5. So our interest is the nanoparticle. It should be dissociated at acidic conditions, and it will release more amount of drug only at acidic conditions. So that I have proven by using my drug release studies. We can see in the cumulative LF release, it is a green line shows pH 7, pH 4, and the red line it shows pH 7.4. So under the acidic conditions, the amount of lumefantrine released is higher than compared to that of physiological pH. So it will be more useful and for the cancer. That is because our cancer cells should be in acidic conditions, so it's the release rate will be higher. So that also I confirm by using my release studies. So these are the other parameters I have checked to prove its efficacy. So this is a HPLC chromatogram. That is at different pH conditions, amount of lumefantrine eluted was checked in HPLC instrument. So at pH 4 only we can get a peak higher. That means amount of drug released in pH 4 only higher. But pH 7.4 and 5, there will be a slight decrease in the LF release. It shows my developed formulation. It comes under only in the range of release will be better in the only acidic conditions. So these are the overall preliminary in vitro characterization studies. After that, we have to move for the in vitro cell lung cancer studies. For lung cancer, we have to check first in cells. That is, cells is we have to check in lung cancer cells. Lung cancer cells is the A549 cells. So we have to check in A549 cells at different concentrations. Final compound is lumefantrine with calcium phosphate cubosomes. So we have to check without calcium phosphate also to prove its efficacy. So I have checked different samples. First I have chosen as a pure drug LF. Second is a cubosome, which is a carrier for my work, and the third one is a calcium phosphate, and fourth one is a without calcium phosphate formulation. So which shows the overall result suggested that our final formulation only shows higher amount of cytotoxicity compared to that of other formulation. So it's proven my final formulation is be better for lung cancer approach in cell lines. So after that, what is the mode of cell death occurs in the cancerous cells? That also we have to study. For that, I have used the method for AOEB staining. AOEB staining is acridine orange etherium bromide staining, in which we can know the cells undergone which type of death, whether it is apoptotic or necrotic. So it is a type of programmed cell death, apoptotic. So my formulations, most of the formulations undergone the apoptotic way of cell death. So it confirms the overall performance in lung cancer cell lines. So based on these results, some of the results also available, but I focused on the main results of my in vitro part. So based on my in vitro results, I have published one research paper in chemistry and physics of lipids with an impact factor of 3.3. It comes under the Elsevier journal. It is my PhD first research paper. After that, we have to prove its efficacy in animal cell lines for in vitro models, in vivo models. Okay, so in vivo models, I have chosen the Cubosome mice. It is a research grade mice specifically for available to check the anti-cancer effects. So it is we have to get approval for that. For handling animals, we have to get approval from Institutional Ethical Committee and the CPCAC. After getting a proper approval, I moved with animal studies, the in vivo part of my animal studies. So first is the animal will be a normal only. We have to inject with the lung cancer cells to make them lung cancer. So here the cells I have used is A549 cells injected to the Cubosome mice. So after the particular time, the lung cancer will be developed in the animals. So that it will take 1 or 2 months. It depends upon the what dose we are providing to the animals. So for my research work, it takes about 2 months to develop lung cancer in animals. So development period. After the development period, we have to check whether the lung cancer is formed or not. For that, we have to sacrifice the mice and we have to take the lungs and you can take a photograph. That we can show in A and B of figure 3. That is first one shows A figure shows a normal lung cancer, normal cells, that is without lung cancer mice. Then second is with lung cancer. In first figure, we can see the regular alveolar structure with non-disturbed nature. But after the lung cancer development, the lung morphology is totally changed and it contains white nodules. That is it's an indication of formation of lung cancer. So further, I confirmed the formation of lung cancer. I have proceeded with the testing studies. Testing studies is nothing but we have to give samples, that is our test samples, pure lumefanine, pure cubosomes, and lumefanine with LS and without cubosomes. So different samples I administered it to the different animals and I have checked its efficacy. After the treatment period, it is a downside figure. We can see the normal and control and the treatment groups. In case of normal, it shows a normal structure, but in case of control means it is a lung cancer induced group. So here we can clearly visualize the lung cancer is developed by larger size of lungs. So it is an indication the lung cancer is developed. So after that, we have to check the weights, individual weights of the lungs. So normal lungs shows less weight. In case of control, that is lung cancer developed lungs shows a higher weight, that is 3.9 mg. After the treatment, the lungs weight should become less. Then only it's an indication our drug is work out and it reduces the tumor progression. Thereby, the lung size will be reduced. So it is an indication, it is an overall correlation indication. The size is reducing means our lung cancer is reducing or somewhat preventing our formulation. Our formulation prevents the lung cancer. So this is the overall study of research work, in vivo research work. Based on the in vivo research work, apart from this, I have carried out a lot of studies, that is hematological parameters, that is RBC, WBC counts. By measuring these levels in the lung cancer animal models, we can confirm the tumor progression is reduced or not. And actually, I checked for the hematology analysis and biochemical parameters also I have checked in animal samples. With all these data, I have published this research work in European Journal of Pharmaceutical Sciences with impact factor of 4.384. It comes under the SCA journal. It is a core journal for pharmaceutical applications. It's a core journal. So this is the in vivo part of my research work I have published in this journal. So in vitro part I have published as a separate journal, and in vivo part also I have published as a separate journal. So coming to the another research area, that is my ICMR research fellow work, that is resveratrol quercetin dual drug loaded nanoparticle system for the treatment of lung cancer. First area is specifically focused on lung cancer, but this area is specifically focused on colon cancer. Hereby, I have developed a nano formulation which contains both drugs in a form of liquid. So resveratrol and quercetin. Maybe students not aware about this resveratrol and quercetin. It is a plant-derived drug. It's already available in grapes and fruits. So it is a normally available drugs only. So by using this normally available drugs, we can develop a nano drug delivery system for the cancer. So this also I have designed a formulation by using nano formulation techniques, that is QRNP is a developed formulation. Then I have checked its particle size and the zeta potential, and I have developed the one method, that is HPTLC. HPTLC is instrument high performance thin layer chromatography, and that's a thin layer chromatography which we can monitor how much amount of samples present in the formulation based on our standards. So by using the quercetin and resveratrol standard, I can estimate how much amount of drug present in the final formulation. So that's we can confirm by using a HPTLC. Here I have developed one specialized method which can be used to detect the presence of quercetin and resveratrol in some extracts. So here I have chosen this as Sesbania grandiflora. It's one type of plant. In the plant extract, how much amount of quercetin and resveratrol it's present in the extract, that I have analyzed by using HPTLC. Then QRNPS in formulation also, how much amount of drug is present, that we can be analyzed by using HPLC. So this is a newly developed method. I have developed this method, and I have published this method in one paper, that is a combinatorial analysis of quercetin and resveratrol by HPTLC. It is a basic type of TLC only, that is thin layer chromatography, but it is an auto sampler, so it's a high performance thin layer chromatography. So this is an instrument which I have used for the high performance thin layer chromatography. So it's basically on the plant derived formulations. So this work is published in the Natural Product Research with the impact factor of 2.1. It comes under the category of Taylor and Francis journal. So this is ICMR research fellow work. After that, before the PhD work, I have worked as a project assistant in the center called National Facility for Drug Development. In the period, I have carried out one research work that also for drug delivery applications only. So in that, I already told that inorganic type of nanoparticles. So this material comes under the organic and inorganic type of nanoparticles. That is, zinc is a metal ion, and 2-methyl-imidazole is a organic linker, and the metformin is a drug which I have chosen for this study. So each and every researcher, we need a drug, we need a carrier, we need a disease. So zinc is a metal, and 2-methyl-imidazole is an organic linker. The drug is a metformin. It is in a chemical synthesis process, it forms a metformin-loaded zeolitic imidazole framework. And this work also is based on the pH sensitive mechanism only. That is, at pH 7.4 how it works, and pH 5.5 how it works. The diagrammatic representation it shows at pH 7.4 the drug will be the inside the carrier only, but at pH 5.5 the drug will be released because the dissociation of ions at this acidic environment. So this is a schematic of zeolitic imidazole frameworks. It comes under the category of metal organic frameworks. So further, I confirmed the formation of this nanocarrier. I proceeded with my in vitro characterization studies as explained earlier, that is particle size, TEM, SEM, all characterization techniques. After that, I have confirmed the anti-cancer activity in same A549 cell lines. Here we can check the particle size in nanoparticle analyzer and HRTEM also. So it clearly shows the porous structure. So the advantages of porous structures is we can accommodate large amount of drug within the pores, and the samples will be released at acidic environment only. In case of physiological pH, it will be remained within the pores itself. So it is advantages of this type of nano carrier. So this unique carrier. Recent lot of research works are going on the this area, that is zeolitic imidazole frameworks, for other applications also. For catalytic applications and environmental application. Here I have focused this carrier for drug delivery applications, and these are the overall results of project assistant work worked in the National Facility for Drug Development. So this work is published under the materials technology, that is advanced performance of material. It's also the impact factor with a 3.84. The publisher is Taylor and Francis. So based on my overall research experience in cancer, I have published a review paper specifically focused to the drug delivery system, that is recent progress in stimuli responsive intelligent nanoscale systems for the different type of diseases. In this review paper, I have specifically focused towards a pH sensitive, because of most of my research publications is focused mainly on pH sensitive. So overall, I concluded all what are the type of pH sensitive systems are available. So I consolidated all, and I have published as one of the review paper that's under the current drug target journal with impact factor of 3.4. It comes under the Scopus journal, and the publisher is Bentham Science. So my area of research is mostly on the cancer only, but I have involved in various collaborative works, co-author works with my team. So another area of research works which I have carried out is rheumatoid arthritis. So this also comes under the category of drug delivery only. My area is cancer, but it is another area, rheumatoid arthritis. It is a joint disease, joint pain disease. So by designing a formulations with methotrexate as a type of drug for rheumatoid arthritis. These are my co-author works. So I supported for this research work. This also comes under the category of drug delivery applications only. After that, I have carried out a lot of co-author research works. That is, I have contributed some parts of the research work in method development part, and some analysis which I have done. These are my co-author research works. So overall, these are my research works. So I want to thank the Society of Chemical and Synthetic Biology for providing the opportunity to talk specialized topic, that is my area of research, nanoparticle for drug delivery systems. So I extend my sincere thanks to Dr. Madan A. Senthil, Chairman, Ratnam Group of Institutions, and my mentor of my research project, the DHR project, Dr. V. Nagaraj, Ratnam Technical Campus, Professor and Department of Biomedical Engineering, and also he was Director of Operation of Atal Incubation Center, RISE. And then I have to tell my sincere thanks to my doctoral guide. Because of her only, I have accomplished a lot of research works with a good position. So I acknowledge my research guide, Dr. Rukmani, Professor, Department of Pharmaceutical Technology, and Director, Center, University College of Engineering, Anna University, Tiruchirappalli. These are the funding agencies I want to acknowledge. For my current research work, that funding is provided by the Department of Health Research, and the host institute I thank is Ratnam College of Ratnam Technical Campus. And for my ICMR research work, I thank Indian Council of ICMR Research Work for providing SRF fellowship for the period of 3 years. And I thank AICTE for the GATE fellowship for my PG studies. And I want to give a special thanks to Anna University, Tiruchirappalli, which supports more facilities. The facility centers available at Anna University, Tiruchi is National Facility for Drug Development and National Facility for Bioactive Peptides, and the Center for Excellence in Nanobio Translation Research. So with this supports only, I have carried out these much works. And now I am being a young scientist in the Department of Health Research. Thank you all. Thank you for all your support. If any questions, you can ask.
Thank you, Dr. Vaidevi. So now the session is open for questions. Actually, Dr. Vaidevi, since we have made it in the live stream, so most of the participants viewing through the YouTube. So there are only limited participants in the online itself. Okay.
Turn right toward Milikurichi Road, then turn.
Okay. So with this, just wind up our session. So thank you, Dr. Vaidevi, for your wonderful talk and lecture. On behalf of society, our sincere thanks to the participants and the speaker for the today's session. Thank you. Thank you all.